Hi everybody, here is another example of how we can regulate the activity of an enzyme through an activator, but this time it is not going to bind directly to the active site. Instead, it's going to bind to another site away from that active site. It's going to bind allosterically to another site on the enzyme. So in this case, same deal. We have, without that enzyme in place, these substrates, or this one substrate that will hopefully be hydrolyzed into two separate substrates, that is not going to fit. That is not going to fit in that active site. We need some help, and that's where the activator comes into play. Now, when the activator comes in, again, it is not going to the active site. It is going to bind to another site elsewhere on that enzyme, away from the active site. This is what we call allosteric activation. And I'm going to advance the slide.
All right, so I've just traced this over, so I've changed this now, and you can see here that we've got the activator. And once this activator has bound to this allosteric site, you can see it's not bound to the active site here. It's bound outside of that active site. It also changes the shape of the enzyme, and now this enzyme is activated because when we look at how this substrate is able to fit in there, you can see now it fits in like that. Fits in perfectly, and there it is. It's done its job. This enzyme can now catalyze this particular biochemical reaction and take that one substrate and turn it into two. You can think of this as kind of like being a hydrolysis reaction, for example, and you can now see that how an enzyme can also be activated through an allosteric site like this, and not necessarily always have to, you don't always have to have an activator bound to the active site. So there's two options there. We showed you in the previous video how an activator can bind to the active site and allow that enzyme to be turned on or active, or you can have an activator binding on to a site allosterically, meaning that it's a site outside of the active site at a different location on the enzyme, and that allows it to also activate this particular enzyme. So there it is, the two sort of different ways that we can activate enzymes through the active site or through an allosteric site somewhere else on the enzyme.
Show you what that then does. When that activator then comes into that activator site, it then changes the shape of the enzyme, and it is because of that change in shape of the enzyme that this substrate can then go in, fits in nicely like that, and that reaction to split apart this substrate into two different molecules can take place. That reaction is catalyzed, and we have one substrate there, another one there, and thanks to the activator, which has activated this enzyme, we can make that biochemical reaction happen. Again, activation of an enzyme, but through an allosteric site, another site away from the active site on the enzyme, and there it is there.